So we are now at the stage where we have finished our intron and exon processing. And so we have my all exon transcript. And we are about to run into a small problem. And that small problem is that eukaryotes have defense systems. Right? One of the biggest problems facing eukaryotes is viruses. And viruses will insert their own genes into a eukaryotic cell and get the eukaryotic cell to follow viral gene instructions and basically shut down that cell and turn it into a virus factory. And so eukaryotes defend themselves against our viruses by having cytoplasmic DNAs and RNAs enzymes. And these chop DNA and RNA to bits. The idea being, if a virus puts its genome into the cytoplasm of the cell, these enzymes are going to chop that DNA and RNA to bits, right? This is defense against viruses. But RNAs and DNAs, they're not capable of recognizing viruses versus the own cell's mRNA. So basically,
here's some results.
We need protection against this. We need to be able to prevent our transcript from being destroyed by the eukaryote's own defense systems. So, there are two basic structures that provide us our defense. The first is the 5' cap, right? So the 5' cap gives us two things. One, defense against our DNAs and RNAs, and two, it's a ribosomes bind here chemical. So ribosomes will be able to attach to our transcript when that 5' cap is present. So this is our 5' cap. Now, what do we have to protect us from DNAs or from RNAs on the other side here? Because as long as it's exposed, it's unprotected. Well, we have a whole string of nucleotides, right? And ready, it's A. Oh, you getting perhaps a guess at what nucleotide this thing is made of, right? So attached to the 3' end is the poly A tail. The poly A tail does not prevent this transcript from getting chopped, right? It does not prevent chopping. Instead, it's chopped first. So, while this transcript is running through the ribosomes, over here we've got getting chopped to bits. So, This actually is pretty cool because it provides us with a fun little bonus ability, and that is a small amount of homeostasis on how many times this transcript will get used, right? Because click, 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 chop, 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 chop. And then once we start chopping our transcript, the transcript, like the first chop, right? Chop here and unusable. So as long as we're chopping poly A tail, we're golden. Their transcript is functional. But once we chop into the transcript, it's dead. So the longer the transcript is in the cytoplasm, the more times it's transcribed, or it's translated at the ribosome, which means the more protein we get. So a longer tail, a longer poly A tail, means more time that transcript is functional. So this is a fun bit of homeostasis. If you don't need a ton of that protein, the poly A tail is going to be shorter. And if you need a lot of that protein, the poly A tail is going to be longer. So that's kind of cool. A little homeostasis bonus in addition to protecting from our chop 'em up enzymes. Do you have to know DNAase and RNAase as a term in your Bio 1 course? Probably not. But, ta-da! Now, once you have all exons and you have a 5' cap and you have a poly A tail, congratulations, you have eukaryotic mRNA. And transcription is done. Which means the next step in our Biology Bite series will be translation.